most of the times, if you see that when the students appear for these kind of entrances, that could be IIT JAM, that could be GAT-B or TIFR related exam, or it could be for PhD exams like CSR NET or Debasish. So sometimes students will prepare for the exam over 6 months or over 1 year. But just before the exam, they have two things in their mind. First thing is, somehow they know from their seniors or from their teachers that they have to practice or they have to solve the previous year's questions. So eventually they will do it because previous year's questions are available in many open sources nowadays. So they can easily access to those previous year's questions. But in addition to that, they also know that they need some, a similar kind of questions, which will not be previous year's questions, but which will give them the similar flavor to the previous year's questions, which is called actually the model questions or similar kind of thing. Okay. So in fact, if you talk about any entrance examination, whether it is CSIR or IIT JAM or any entrance examinations, both the questions are extremely important. I mean, students should be completely familiar with the previous year's questions as well as new questions, I mean the model questions. Now, what we did actually in this book, that we sorted out all the questions across all the previous year's entrance examinations. That includes all the major exams like CSIR, DBT, IIT JAM, GATE, then GAT-B, then previously JNU used to conduct that exam, JNU MSc for Biotechnology. So like that, we actually compiled questions from almost more than 10-15 different varieties of questions. And for over the, you can say more than 10-15 years also. So we had a huge compilation of questions of almost more than 10-15,000 question papers. Now, from those 10,000, 15,000 questions, what we did, we sorted out all the questions because we knew that, let's say someone has already finished biochemistry part. Now he is ready to take questions on biochemistry. So we screened the questions and we put up the questions under biochemistry, that too under different subsections, like biochemistry as a whole. But students, let's say someone is ready with only protein biochemistry. So we kept questions for protein biochemistry. Let's say someone is ready with nucleic acid chemistry. So we kept questions for nucleic acid chemistry. So similarly, we subcategorized all the questions according to the each and every subsections, which will be really beneficial for the students, that once they're ready with any chapter, they can face the questions and they can test their knowledge, how far they stand and how far they need to prepare just before they take the final call. Actually, the motivation comes from the students, of course, because during my PhD time, I realized that there is a need of some special preparation for this kind of entrance examinations. Because if you look at the way how students study in their BSc as well as in MSc, and the way these exams are conducted, there is a certain degree of differences in those question patterns. So someone who is aspiring for MSc or aspiring for PhD, they need to have extra preparation from their ongoing BSc as well as MSc. So what I did, I started giving this kind of coaching since the days of my PhD life onwards, because almost on every Sunday I used to go out of my IIT campus and I used to travel a little bit so that I could cater some students. And to be honest, when I was teaching those students, I could realize that they need some book, because in the teaching program, what happens, you got some limited time, let's say 2 hours, 3 hours. But to be honest, when someone is preparing for this kind of heavy, weight exam, so someone needs to prepare for a longer period of time. So as a teacher, you can get hardly 2 hours or 3 hours to teach your students. But if you look at the student's point of view, they need ample amount of questions for their preparation. So he doesn't require the presence of his teacher every time in front of him, but he needs a supportive, material, supportive, preparatory guidance, so that he will keep on practicing the questions. So I realized that, and I understood the need of the students. So eventually I came across, with the different publication proposal and started writing the books. Yeah, my experience with scientific publisher Jodhpur is extremely good. Like I could see the number of sales, the response from the students is really good. In fact, I was expecting that the book should reach every corner of the country, because this book has a tremendous potential for all the students of life science, whether it is in BSc, whether they are in MSc, and that was reflected in the sales. Yeah, if you look at the subject biotechnology, it has a huge potential, to be honest. It's not like since I'm in the biotechnology domain, I'm trying to defend my subject. It's not like that. I can tell you few things about the other science subjects as well. You will see that biotechnology is a subject which is merged and which invites all the other scientific students, like students from physics, chemistry, math background. They are working with biotechnology. So biotechnology has a huge scope of working. In fact, the students of pharmacy background, they also come here to work. You take an example of the vaccine, the recent COVID incidents, the vaccine. If we talk about the vaccine production and the mindset it requires from the beginning to the vaccine development, it requires the help of computational biology approach, which is so-called known as bioinformatics. And in this area of bioinformatics as well as data science, there are many students who are from non-life science background, like students from physics, students from chemistry. They are working on this area, bioinformatics as well as computational biology, because they are the base of this. If you are talking about any new molecule production, whether it could be vaccine, whether it could be any drug molecule, anything, because that is the base of the discovery. Once the base is developed, once the base is identified, then biological samples will start into play, and they will think about developing that molecules through different assays, through different clinical trials and all those. And finally, it has to pass some regulatory affairs, so that with the government's approval and all, so final molecule will come into the market. So this is just an example I'm telling you, where you will see the application of many different science subjects, and they are coming into the field of biotechnology. Now, I'll give you one more example, which recently the world has already seen. Gene editing technology. This is one of the biggest, you can say, discovery which has happened recently, maybe 4 or 5 years before. So what happened actually? You will see this. If you look at the concept of DNA, so our body is actually controlled by DNA. But the thing is, DNA doesn't control anything. DNA encodes a protein which is called a gene product. Okay, so this protein controls everything. So everything is actually controlled by the protein, but the protein is actually encoded by a gene. Actually, to be more precise, it is encoded by a gene. Now, sometimes what happens, let's say some protein, the gene product is not good for us, but which is expressed by the cell. So you don't want the production of that protein, but still it is expressed. So what happens actually? Basically, it is a part of our body, but we don't want it anymore. So sometimes what happens, you need to edit the function of that gene so that that protein is not produced, or protein should be, degraded, or the expression of that protein should be less. So there is a process called CRISPR-Cas9 gene editing technology, which is very famous, and in fact, the scientists got Nobel Prize also for this. So this CRISPR-Cas9 technology helps gene editing process much simpler for the life science scientist as well as the biotechnologist, you can say. So through this approach, you can modify any gene, so that, let's say, a gene which you need for more, so you can eventually fine tune for the better expression of that protein, and the gene, let's say, you don't need it, you can delete it out as well. So editing means both of you can do. You can go for switching on or off. So this will make the life science job much easier, as well as better function of the cell, as well as tissue, as well as organ, as well as the healthy body.